actual scientific name is Homo sapiens, right? It's a genus and the species. And so I bring that up because they're going to reference it, but it's not something that you are going to need to memorize or know, okay? So you're not going to be tested on anything to do with taxonomy. You're not going to be tested about memorizing their kingdom, their domain, their phylum. Like those big words aren't something that you're going to need to remember. But I wanted to bring it up because it does get referenced to in these future lessons. We have a couple of really interesting topics that we're going to be tackling. And so we're going to start off by understanding in the lesson, in the order, it's going to talk about things like virus and protist and bacteria and archaea. And I started to put together a lesson that was going to go over all of it, but I kind of stopped and just decided to focus mostly on viruses because we're in a pandemic due to a virus. So I have a video for us to watch and a couple of notes that I want you guys to really pay attention to. So that way, small group, we're going to learn about the coronavirus specifically. So we're going to learn about viruses in general, and then we're going to learn about why COVID and the disease that's caused by the coronavirus is so different than anything we've experienced before. So that's what we're going to do in small group this week. And then just remember that it goes from like virus and protist, bacteria and archaea, and then it jumps into digestion, and then it jumps to human digestion. And you're going to be doing a human digestion lab at the end of the week. And I have the video that I went over yesterday in the lab yesterday. So I'll be posting that today, but just wanted to give you guys kind of a preview of what this week is going to look like. Questions on that so far? If not, no big deal. Okay, let's get started. So like I said, I only have a few slides. I do want you guys to be taking notes on your own. So I don't have a note sheet for you to fill out while you are while you're following along. So just make sure that you have a piece of paper and a pencil next to you, because we are going to talk about viruses, and we are going to talk about the two different cycles that it goes through. There's two different ways that the virus can reproduce, and we're going to go over that. Some really interesting things about viruses. Not like I'm excited about this, because I'm not, but it is incredibly interesting to learn and read about the complexities, because there is a lot of different aspects when it comes to something like a virus. So there are, oh no, I didn't fix that. Let me delete this real quick because it's going to bother me. So I really did not, I thought I had finished this. So really, let's focus on virus and bacteria. So virus and bacteria. So a virus is, give me a second, a non-living particle that reproduces by invading and using the mechanisms of a host cell. So obviously, we're going to ignore these other three. So I really want you to focus on this definition. Couple of things to pay attention to. It's a non-living particle, which is crazy to think about. Viruses are not considered alive. And we're going to talk about that in just a moment. But it's crazy that it can cause so much damage for something that's not technically does not meet the criteria to consider something to be living. So let's look a little bit closer. So we talk about in the beginning of this entire school, right? In quarter one, one of the very first things that we learn about are the criteria for what is considered alive, right? There were 7 characteristics of what we consider alive. And in order for an organism to be considered living, it needs to meet all of those criteria. Well, viruses are very interesting because they don't meet all of that criteria, which is why there's an argument to say that viruses are not alive. So something like viruses are not cells. They're not cells. They're not made up of cells. They're made up of specific like machinery that create the virus, but they're not considered cells. Interesting. They do not grow. They do not multiply. So they don't have cells. They can't go through mitosis. Remember, mitosis is how things grow, right? So they don't go through mitosis. They don't grow. They don't get bigger. Okay. So viruses don't grow. It's a pretty big part of something that's considered living. They do not take in energy from the environment. Remember, we talk about metabolism. We talk about needing to eat food and create sugars, either by creating it themselves or obtaining it through eating other animals to get that energy. They don't take in any energy, and they don't metabolize, so they don't necessarily need food to survive. So it's interesting. These are all characteristics that we specifically say are needed for an organism to be alive, yet they don't meet 4 of the 7 criteria. So we don't consider viruses to be living. It's wild. But yet they do have a way that they can reproduce, and they can survive, but they need a host cell to do that. So we're going to talk about the lytic cycle. So there are, like I said, and there's going to be an image coming here after that's going to give you some very specific imagery, like some visuals to help. But let's start with the first step. So in order for a virus to survive, it needs to attach to a cell. So it cannot reproduce on its own. It needs a host cell in order for it to reproduce. And so the very first thing is the virus is going to attach to the cell. The video is going to show you how it does that, and the image will as well. There's a specific thing that needs to occur in order for it to attach to the cell. Then it goes through a phase called injection, which means the virus literally punctures its way into the cell. So it's on the cell surface, okay? So it lands on the cell surface. It goes through that cell membrane, breaks through it, and injects its own DNA. So we call that the viral nucleic acids. I need to fix this real quick. So we have the viral nucleic acids, which means the viral DNA. Remember that nucleic acid is the DNA, right? Nucleic acids is one of those groups of macromolecules, and it is specifically representing DNA or RNA. So there are 2 ways. That's why there are 2 ways that cells can be considered, or excuse me, viruses are considered, and it's a DNA or RNA. And we should be familiar with those 2 terms. So if we say RNA, what does that exactly mean? And if we say DNA, what does that mean, right? So making sure that we understand the type of virus we're working with, but regardless of what it's inserting, it's putting in its own genetic information into the cell. Okay, so it just kind of puts in the DNA. Now, replication. So now it's given its DNA into the cell, and the cell doesn't really have a way to determine if it's its cell's DNA or if it's the virus's DNA. They just see DNA. So what happens is the cell starts to form, take that DNA, and it just starts to create those virus parts. It just starts to build it. Starts to build the protein, right? Viruses are a protein. So it's sneaky in the sense that it doesn't need energy because it doesn't need to do anything. It just needs to land on a cell and then give it its DNA, and then the cell's going to do all the work for the virus. So then they put the virus DNA into your cell, and then your cell is going to go through the process of protein synthesis. It's going to go through DNA to RNA, and then mRNA to the ribosomes, and the ribosomes are going to crank out those proteins like it's supposed to. It's just doing its job. So then there comes this assembly part. So sometimes the virus can give the directions, the DNA, right, the codes to create the different parts of the virus. And then during the assembly phase, they kind of find each other and they connect and reassemble and create a new virus. And then you've got the release. So as these viral DNA are getting cranked out through the ribosomes, and as the ribosomes are creating the machinery, like all the parts that the virus needs, the virus comes together and connects it. Then you've got a lot of viruses in your cell to the point where the cell can no longer contain the pressure. There's pressure being pushed against the cell membrane. And then eventually what happens is it starts to burst. Your cell actually bursts open. And then the virus goes and is released into your body, into the bloodstream or wherever it is, and starts to attack host cells, new host cells, and the process repeats itself. So here is a visual. Okay, so this is just the lytic cycle. And again, there are two different cycles. There are two different ways the virus is going to be able to happen, or it's going to be able to take hold. And so the lytic cycle is probably the fastest one, where, you know, if we're really trying to get as many viruses, if their goal is to like infiltrate, right, and find as many host cells, the lytic cycle is going to be the way that they're going to do that the fastest. Okay, so again, we've got the same steps. So we've got the host recognition. So we have a virus is coming up to a cell. It lands on the cell. So it's the same cycle, just maybe different vocabulary, right? Yes, they do look like aliens. As a matter of fact, there's some movies that base their aliens to be like viruses. I know. And then we've got this absorption, right? So again, different vocabulary, same idea. They land, they connect to the cell. Again, we're going to see how the viruses connect to cells. And then they inject their DNA into the cell. And then over here, we have the DNA starts to become the cycle. So one thing, this is specifically showing you what happens when it lands on a bacteria cell. So like when viruses infiltrate bacteria, remember that bacteria are prokaryotes and they have one single circular ring of chromosomes. They don't have multiple like we do, right? So then they become circular as well. And then during the replication, this is when we get the new phage. So phage is the different parts of viruses, excuse me. So bacteriophage is viruses that infect bacteria. So phage are the different parts of the virus. So then we have the different parts. So just like I said, they actually have the DNA to make just the different parts of it, and then they assemble. So then they come together. They're like, oh, there's a leg, there's my body, and there's my head. Let's bring you all together. And then they assemble, and then when there are too many and they create that pressure against the cell membrane, they cause the cell membrane to break, which means, which is with this lysis. Lysis is another fancy sciencey word for break. And then they are released and they go to find new cells to infiltrate. So the flood from, there you go. So many good connections, Emmett. Yeah, maybe there's something into it. Maybe the creators of all of those characters had a common idea in mind of what they wanted to model their aliens after. Good connection. Okay. Are there any questions about the lytic cycle? We're going to go into the lysogenic cycle in a moment. Any questions on the lytic cycle? Okay. So when we have the lytic cycle, we also have this thing called the lysogenic cycle, which is they're interrelated. So they're not one or the other. I have them separated just so we can understand the two different parts, and then I'll show you what it looks like when they are entwined, okay? So we just learned about the lytic cycle, which was pretty straightforward. It's an immediate response. It's immediate action where the virus duplicates as fast as it can, and then we see the results of that by a lot of multiples of the virus and, going to the host cell. Now, the lysogenic cycle is actually a really sneaky thing that these viruses can do. It's kind of like the sit and wait approach. And this has to do with gene expression. So I'll talk about that in a moment. But it's going to be similar but different. So the beginning is the same, where they're going to attach the virus, right? The virus attaches to the cell just like it does in the lytic cycle. And then it's going to inject their viral nucleic acid, so their DNA or RNA, depending on the type of virus it is. And it's going to inject it into the host cell like it does in the lytic cycle. Now, this is when we start to see it be a little different. So in the lytic cycle, right, in the lytic cycle over here, it puts the DNA and it's right away, it is separate from the bacteria's DNA and it just is getting replicated, okay? But in the lysogenic cycle, now what we have happening is the viral DNA actually coils next to the bacteria and it actually embeds itself, it embeds its own DNA to the DNA of that bacteria cell. Okay, and we're talking specifically about bacteriophage. So I'm specifically talking about viruses that infiltrate bacteria. This does happen in eukaryotes and organisms, but it's going to be it's not always going to be exactly the same, right? So there'll be a couple of changes when it comes to humans and animals. So viral DNA coils next to the bacteria DNA. So then what happens is it actually becomes part of the cell. So every single time the cell replicates, it also replicates the DNA for that virus. But the virus isn't activated yet. It's dormant. It's like asleep, okay? And then once the environment calls to and it's activated, then the viral DNA is going to move into that lytic cycle, the one up here. So, you know, we talked about gene expression in one of our earlier units this quarter, specifically talking about how genes can be turned on and how genes can be turned off. And you can have the genes and then it awakens. Exactly, Christian. And a few of you are going to love some of you guys are absolutely going to love my analogy that I thought of last night, and some of you guys are going to be upset if you've never seen it. So just give me a second and I'll get to it. I'm like really excited because I know like Emmett's for sure going to like it. So give me one moment. But exactly like what we talked about with the gene expression, how you can have a gene, but it doesn't get turned on until either the environmental conditions are right or time goes on, or you're in a specific process of your body, right? So things like when you go through puberty, that's a whole process of your hormones being activated, genes are being turned on, causing all these different types of developments. Same thing with Alzheimer's. You can have the Alzheimer's gene, and Alzheimer's, if you're not aware, is this condition that attacks the brain. You basically start to forget, and your brain, your memory is affected. It's really hard, and eventually your like actual physical movements will be forgotten. So it's a pretty hard disease. My nana had it. My grandmother growing when I was younger had it, and it was really hard to watch. But that's an example of a gene that's turned on later in life. Right? That's a gene that turns on when a specific environment is right for the conditions, which is the same thing that happens to the lysogenic cycle. The DNA is embedded, but it doesn't turn on yet until later on. And once it becomes activated, then it goes into the lytic cycle where the process duplicates, it starts to create the machinery, it starts to assemble, and then it breaks the cells open. Let's take a look at this image before I go in. So this is the one where I was telling you how at first I wanted to show them separate. And then once we get here, I'm going to show you how they are intertwined. So we started here with the lytic cycle. Okay, so viruses that have this specific pattern are going to stay on this side of the cycle. So we've got our phage DNA that's going to bind to the cell, put its DNA into the cell, and then it's going to start to create the new phages, right? The new parts of the virus until it pops open the cell and then it goes on to the next one, right? And then here's the lysogenic cycle. So this is kind of where we start to see it be a little different, right? So this is where it goes in, it attaches itself, but then it integrates into the host chromosome. So here's a good image. You've got the DNA from the virus and you've got the DNA from the bacteria. And when it goes into the lysogenic cycle, it starts to put itself into the host's DNA. And then what happens is that as the cells divide, and remember, this is bacteria cells, so they have a different way of dividing than humans do. We go through mitosis. Bacteria doesn't have enough chromosomes to need to go through that. So they just literally copy their DNA and split off. That's all they need to do. That's why bacteria can multiply so quickly. And so when the prophage, which is the virus, DNA is copied when the cell divides. So again, the DNA gets copied whenever the bacteria cell divides. So then we have something such as exposure to stress, such as UV light, can trigger the excision from host chromosomes. So it can trigger that specific DNA being cut out. Okay, that's what that means by excision, right? So then it means that that specific DNA gets snipped off, and then it goes into the lytic cycle, where it starts to go ahead and go through the process of building, creating, and going through and bursting open and starting over. So that is the general basis. So here is my amazing analogy to the lysogenic cycle, because I think the lytic cycle is pretty self-explanatory. Virus goes into cell, puts its DNA into the cell, creates copies. When the cell is creating copies, more viruses are made. Pop goes the cell and then finds its new host, right? The lysogenic cycle is a little more difficult. Now, I have to preface this because it is a spoiler alert, but how many people have seen Guardians of the Galaxy Volume 2? Yes, 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 yes. It's a good movie. If you haven't, I highly suggest that you watch it. It is like one of my favorite series of the Marvel, of the universe. Okay, how many people have not seen Guardians of the Galaxy Volume 2? Okay. Okay, next question. How many people who have not seen it plan to see it? And if I move forward with my analogy, it will ruin the plot for you. Okay, I don't see any responses to that. So I'm assuming I am okay to move forward. Is that right? For those of you who did not make it to class today and are watching this recording, you should skip over this part if you do not want to, spoiler alert. Okay, so for Volume 2 of Guardians of the Galaxy, let's just give some context to those who haven't seen it. So at this point, they've already established themselves as a family. We've got Quill, who is the main character. He finds out in the first movie that he is actually half human and half something else. He never met his dad. His mom died when he was younger, and he got picked up and got abducted from Earth into space or into the galaxies, right? By this whole group of people who were pirates of a sort. And basically, Volume 2, you start to find out who is Ravagers. Thank you, Ravagers. And it is a sad backstory. So then in Volume 2, we start off with them trying to work with another planet by protecting them, and then they get attacked. And then finally, they find out that his dad comes into the picture, okay? And it turns out, this is why it's like a big spoiler, it turns out that his dad is actually a god. He's a celestial, which means that he's not really human. He was created during the Big Bang, or when they don't really have they do have the Big Bang, but then they have the Infinity Stones that came out of that, which is a whole separate conversation. We're not getting into Infinity Stones, but he was created when the Infinity Stones were created. He's basically just a god, part celestial. Thank you, Emmett. Yes. And with that means that he has this control over matter. He can create it. So he actually created his own planet. I'm getting really, really technical in my analogy just because I think I just really enjoyed the movie. So let me try to speed it up. Basically, there's this one part of the movie, right, where the father, the celestial, Quill's father, goes around to all the different planets. Thank you, Ego. Yes, he really tries to He finds his meaning because he's just been wandering alone in life all these years. And so he decides that he wants to take a world global takeover, right? And so what he does is he goes to all of these planets and he plants this seed into the ground that he hopes to use when the environment conditions are right. You see where I'm going with this? So he goes and plants this like plant and this seed into all of these planets around the entire universe, okay? And he realizes though that he does not have enough power to turn these viruses on. So they are laying dormant. They're in the lysogenic cycle. Do you see where I'm going with this? So he goes around to all the planets, putting in these seeds, and then he's also reproducing with some of the people that live there so that he can have an offspring that has his genes. Oh my gosh, so much science involved in this movie. I love it. And then he's hoping that one of his offspring is going to inherit the celestial gene that's going to be strong enough, or that there's enough genes that they can then also power the celestial and the ego, right? And all of those things that are planted into the planets. So they're all dormant until he finds Quill. And Quill has the celestial gene. Now he's able to power and create the right conditions. He's able to create the right environment to turn on that virus. Oh my God, so exciting. So then he starts to use Quill as a battery, as a power source to turn it on. And then all of a sudden, all of these viruses that he planted in all of these planets start to turn on and they start to take over. And his whole plan is to take over the entire universe. And so these things come out of the ground and they start to grow and they start to like kind of suck in all of the things that it comes in contact with, and it all becomes ego. And that's an example of the lysogenic cycle. That it just sits there. It's just dormant. It's asleep until something turns it on and activates it. And then it goes into the lytic cycle, where then it starts to get built. The DNA starts to get put through the ribosomes. And then it starts to create the different parts of the virus until there's so many in the cell that it pops open and it goes into the next host cell and the process repeats itself. And that's my analogy. Tell me that's not like brilliant. I thought of this literally last night as I was getting prepared and thinking about my lesson today. And I was like, what am I going to do to explain the lysogenic cycle? And I love it. I think that I'm gonna get that patented. It is my intellectual property. And if you ever use this example, you have to give me credit. So you have to say, like, even if it's in 20 years, and if you're like, for some reason, having to remind yourself about what the lysogenic cycle is and you go to explain it to someone else, you have to be like, my 9th grade biology teacher, Miss Fierstein, told me, and then present the analogy with Guardians of the Galaxy Volume 2. And if you haven't seen the movie, you should, especially now, because I know it took me a couple times to understand what was actually happening in that movie. But, all right, that was my analogy. Thank you, Logan. I think I pat on my back. I was really proud of myself for that. Okay. All right. So that in a nutshell is a virus. Like, that is when we get sick from a virus, we're getting sick by these things that are infiltrating our cells and causing our cells to pop open. And then that's when it elicits an immune response from our bodies. I'm not signed out. I'm right here. So what I want us to do now, because I really like, I think that watching videos is a really great way to also understand it a little bit better, because, a lot of what we're doing right now with the online program is reading. And I don't know about you guys, but I specifically am very much visual learner, not a virtual learner, a visual learner. And so watching a video really, really helps me understand what I just read. So we're going to watch a video. As you can see, there are a ton. And I'm going to choose this one because it looks cool. And if it's bad, I apologize, but it's only 3 minutes and 39 seconds of your life, and I think. And then let me put it into this. And for those of you who are watching this as a recording, I know the video is not going to play. So take a look at the description of the YouTube video link and click on this video link when you get to this part. All right, so for everyone else that's here live, you can take notes. I always strongly recommend that you take notes, especially if there's something that you're watching and you're like, wow, I had no idea this is like really makes more sense to me. I would write it down, but you don't have to, okay? You don't have to write this down. I am gonna go ahead and put the link of the video into the chat just in case something doesn't get covered. And or if you missed something and you have the video now to be able to go back and watch it on your own time. Okay, so let's go ahead and watch this for the next couple minutes. I just want to specify this is not a video about COVID, okay? Again, small group, we're going to be learning about COVID as a virus, okay? We're going to be learning specifically about COVID, but this one is specifically talking about just a flu. So this is not to do with COVID or anything like that, okay? Just wanted to put that out there. That really was a wonderful sound effect, Spencer. I could not agree more. So that was actually an example of a bacteria in a eukaryote, right? We saw that there was a membrane bound organelles. What I was talking about, excuse me, specifically was the bacteria, which is fine. I mean, a virus is a virus. So let's see if I can find bacteriophage video, because it's still a virus. We saw a little bit more of a complex system. I was teaching more of the bacteria. So here is a video about the bacteriophage that kind of makes it a little bit easier to, what's on here? That was oh man, it's not gonna let me use it. Okay. Well, like I said, that was the example of a eukaryote. So remember the difference between a prokaryote and eukaryotes. Eukaryotes have membrane-bound cells, organelles. So that's why we were able to see the nucleus. That's why we were able to see the different organelles in a cell. Whereas what I was talking about was very specific to a bacteriophage, bacteriophage. So here is another video we're going to watch about that. And this one should work this time. Phage, again, is just another example of, oh, good. Sorry, this is just another example of a way to say virus. So when you say phage, it's, again, a virus, right? Bacteriophage is a virus that attacks bacteria, which is what I was talking about in our lesson today. Okay, so a couple things. Remember, this is specifically for bacteria. And between the two videos, you should have seen, right? If you were looking at if you paid attention to the two videos, what was different other than knowing that one was for eukaryotes and one was for bacteria? What was the difference about the virus themselves? Anyone notice? The shape, right, Spencer? Exactly. So the shape was different. And the shape of the viruses were different, which caused a different way to enter the cell. How did the virus in the eukaryote enter the cell versus how did the bacteriophage enter the cell of the bacteria? What was the difference there? Anyone? Bueller? Bueller? How they entered the cell had to do with their shape, right? So bacteriophage can only really enter bacteria. They talked about how it has a separate relationship with humans and eukaryotes, right? It first just went in and then injected itself, right? Exactly. With bacteria, it just kind of forced its way through. It punctured the cell membrane and put its DNA into the bacteria cell. Where with the eukaryotes in a virus, it was more of like a round with like the different keys on the outside of it, and it got welcomed in, and then it burst open and put its DNA into the eukaryotic cell, right? So I just want to point out those two differences. Like I said, today was a lot about viruses, just because we're literally our life has been taken over by a virus the last year. So I wanted to pay attention to that. The rest of this week's lesson is going to be going over the digestion and bacteria and protist and archaea. So you're going to learn a little bit more of the different types of organisms that are out there, and then how the digestive system watches also. Yes, Christian, I've seen it actually. We are going to learn about the COVID virus in small group. So I actually want to save that video for you guys when you come to small group this week. So we're going to learn about how COVID is different than some of the other viruses that we have come up with, or not come up with, but that we know about, and why this one has been so impactful compared to, let's say, the flu and whatnot. Okay, but that's what I have for you guys today. I hope you guys got a better understanding of viruses and the lytic cycle and the lysogenic cycle. And please come to at least one small group this week. Remember, small group is the same lesson throughout the week, just in different times. And yes, Christian small group is tomorrow. There's one every day for the rest of the week, except for today. And then don't forget to watch the video before you get started with your lab, because I do go over a couple of things and kind of walk you through what you should be doing. The lab for this week is a little different in that you are going to be creating your own procedure. You're going to be choosing your own question, and you have to come up with your own procedure, and you are writing a lab report. So make sure you watch the video, because I go over the different parts of the lab report and the grading rubric and all of that. All right. Thank you all so much. Thank you for joining me on this lovely Tuesday morning. I hope you all have a wonderful rest of your week. See you later.